So very good afternoon everyone, very warm good afternoon and welcome to this session. In this session we'll talk about the few of the experimental techniques that can be used for the nucleation and observation of gas and vapor nanobubbles. So this in the past 2 days we have been discussing about theory, mostly and theoretical framework, that what is how the bubbles are formed, how the phase transition occurs, what states are metastable states, and likewise. In this lecture, we'll focus mostly on the gas and vapor nanobubbles. And as I discussed in the last class also, that these bubbles, they can be defined as ultrafine gaseous domains which exist on the liquid surface interface or in bulk liquid. So these bubbles, they can be formed. Let's suppose that we have again a typical box, and let's suppose that this surface is hydrophobic. Now hydrophobic, it means that it doesn't have affinity to water. So it might happen that if we have a look at the microscopic scale of this surface, there will be a lot of roughness here, lot of troughs and asperities. These might trap some gas molecules, and when we fill in the liquid on top of that, and if the liquid is super saturated, it means that it cannot dissolve any more gas into that. In that case, we can observe nanobubbles here. Sometimes these nanobubbles, they can also be in bulk state. Typically, it's difficult for a vapor nanobubble to be in bulk state, but the gaseous nanobubbles, they can be in bulk state, provided that the gas saturation is so high that all the surface which is available for heterogeneous nucleation is totally filled with bubbles. So in that case, what might happen is, like we have two bubbles very close to each other, and let's suppose that we have very high saturation here. So it might give rise to homogeneous nucleation gas bubbles. This has been observed experimentally, although in terms of theory we don't have much proof how they can exist, because what's stopping the size increase of these bubbles. But experimentally it has been proven that we can have homogeneously nucleated gas bubbles. Now one major concern with these gas bubbles or nanobubbles in general is the stability. These bubbles, if we try to calculate its excess Laplace pressure, that pressure for a bubble having a radius of 100 nanometers, it comes out to be 1.4 megapascals, which is very high. Still, these bubbles, they exist for a very long time. If a bubble is of 1 millimeter, that pressure drops to 144 pascals. Similarly, using the Rayleigh-Presset equation, we can find the time scales for which these sized bubbles should be stable. And for 100 nanometer bubble, the times comes out to be 0.1 millisecond. Yet experimentally, it has been proven that these bubbles, they exist for not only days but weeks and so on. So what is causing these bubbles to be ultra stable? That is still a controversy, and many researchers are still working on this problem. But that is not the main focus of this lecture. The main focus of this lecture is these bubbles exist and how do we observe these bubbles, because they are very small. Now, before that, why do we need to study nanobubbles? Because they have got certain exotic applications. The first one is water treatment and environment remediation. So these gas bubbles, especially gas bulk nanobubbles, they are helpful in the water treatment, and moreover, they are also useful for agriculture and food industry. They have got application in biomedical and medical applications, such as if you want to have a targeted drug delivery inside your organ, then you can use these nanobubbles for targeted drug delivery. Energy application is other aspect where these bubbles are very important. It is being said that, or it is being almost proven by some experiments, that the early nucleation, it occurs because there is nanobubbles on the surface, especially in hydrophobic cases. So in hydrophobic, we have as early onset of nucleate boiling. That is because nanobubbles, gas nanobubbles, they act as a cavitation sites for these bubbles, vapor bubbles, to grow. So this is one application. In energy, there are also some papers who prove that the micro droplets which are formed on a surface, that are also because of the nanobubbles. Because let's suppose that we have a surface. In this surface, there may be some nanobubbles, and if a liquid film is receding on that side, then these bubbles, they try to pin the liquid film, which might lead to the formation of micro droplets here. So that is also one application of nanobubbles. Certain applications more towards nanoscale, they also prove that these nanobubbles, when they are nucleated on the surface, they provide almost zero slip. There's no slip, so we can have a very fast flows, or very controlled fast flows in microchannels because of these nanobubbles. So applications are immense, and as we know more about these nanobubbles, the applications, they keep on growing. One other application of these nanobubbles, or understanding of these nanobubbles, is that when we talk about various electrolysis processes on the electrodes, these bubbles, they grow. And when the bubbles are, these bubbles grow on the electrode, they kind of act as an impedance layer between your electrode and the electrolyte. It means that they stop the reaction. So to, to know under which conditions these bubbles are stable, so that we don't reach to that conditions, it is also important in the case of electrolysis. Immense applications, and as we know more about these bubbles, the applications grow. Now, what could be the challenges in experiments for these nanobubbles? I want to take your opinion on that. Like, what do you think is the major hurdle of these nanobubble observation? I think it is size, right? The size of the nanobubbles, since they are ultra fine, so size could vary anywhere between 6 nanometers to somewhere, I think, around, we can call even 1000 nanometer bubble as a nanobubble, right? And if we talk about light, the wavelength of light is between 400 to 700 nanometers, right? So if I draw the scale here, so I think this side is UV, we have 400 nanometers here. This side we have IR, and we have around 700 nanometers here. And between this, the wavelength, it is varying. This is a light spectra. It means that it is inaccessible to your naked eye, as well as all the microscope which work on light, principle of light, right? Because the light, the wavelength of these light is so large as compared to nanobubble that we cannot observe these nanobubbles there, right? So what could be the other illumination source that we can use? Monochromatic light, how will it help? X-rays is one wonderful example, because they have got very low wavelength. So we can use X-rays for their observation, right? We can use electrons for their observation, because they have very low de Broglie wavelength. So these all things we can use for their observation. The other challenge, in not typically in gas nanobubbles, because they have very long lifetimes, but in the case of vapor nanobubbles, is that they have very short life, because vapors, they are very labile. Any fluctuation to the vapor bubbles, it may lead to either increase or collapse of the bubble, right? So in case of vapor nanobubbles, time is also a very important parameter. At how much frame rate, or at how fast we don't need to acquire the data, so that we can observe these vapor nanobubbles. So we have two challenges, both the length scales and the time scales. But these length scales, it mostly is the case in the case of gas nanobubbles. And time scales, in addition to length scales, is mostly the problem of the vapor nanobubbles. Vapor bubbles, they are both small and very transient. So the basic principles of observation, or for capturing these nanobubbles, is one is observation, one is sensing. Observation means you are able to see the bubbles. Sensing means you are not able to see the bubbles, but you are able to sense if the bubbles are present or not. So we can broadly classify the techniques in observation and sensing techniques for these bubbles, right? So in case of observation, it's you are able to see the dynamics. You get a picture, you see dynamics of the bubbles, you see how fast how the bubble is growing, how the bubble is shrinking, and all these things. In sensing, of course, you are able to sense if the bubble is growing, if it's if it's collapsing, but the real picture is not there, or we are not using any illumination source for them to observe. Examples of observation based techniques could be TEM. Now TEM is also known as a transmission electron microscope, and these TEMs, they use electrons in place of light for illumination. And as we know that in the case of TEMs, your wavelength, the de Broglie wavelength is very small. So if it's defined by h by p, h is a Planck constant and p is momentum, and further it can be written as 1.227 by under root of energy. This is in electron volts in nanometers. That is the wavelength of electrons. So it means that if we are able to provide a voltage difference or accelerate electrons at a sufficiently high voltage, then this would be in picometers or even smaller scales. So from a picometer range illumination source, we will be able to observe the nanobubbles, right? So that is the main concept behind observation using the TEM. In sensing, what we do is that we have two distinct methods. One is the AFM. AFM is called the atomic force microscope. What we have is that we have a sample, and using a probe whose tip thickness is in scales of nanometers, we just raster scan our sample, and it basically gives us the profile of what the system could be. Example, if I take the AFM of this desk, we may get all the roughness of this desk on using the AFM. So AFM, if we have nanobubbles, of course we can use this AFM for the observation, but one thing is that AFM, it can have some certain artifacts, because nanobubbles interfaces with soft matter, and as the distance between the two surfaces it decreases, there could be repulsions there. So in AFM we have two different modes. One is your amplitude mode, one is your frequency mode. So we have to see what information do we want, so that we use the appropriate modes for the sensing of these bubbles. The other sensing technique is called the resistive pulse sensing. Now resistive pulse sensing, this technique became very famous possibly around 2011, 2012, because we were able to detect DNA's profiling using this resistive pulse sensing techniques. So what this technique is basically based upon, very briefly I'll tell, later we'll explain in detail. So what we have is we have a very small nanopore, and then we have two tanks. Of course I'm using the word tanks, but the volume of these tanks is very small. And these are placed in such a way that these two tanks are connected only through this nanopore. And then we fill this tank with an electrolyte. Now this electrolyte, it has some certain ions. If we give a potential across this nanopore, what we can see is that there would be the movement of ions. All the negative ions would travel through this side towards this positive electrode, and all the positive ions, they would travel towards the negative electrode. And this kind of creates a lot of joules heating inside the nanopore. This heating is very localized. Of course, it depends on how big nanopore we are using. And this localized heating, it can cause the bubble nucleation. So when the bubble nucleates in the center of this nanopore, it kind of blocks the channel. There is a decrease in the flow of ions when the bubble is nucleated there, which means that if we capture the signal of I versus t, where I is current and t is time, we can see that usually the signal would be high, but when the bubble is nucleated, the signal drops. And then when the bubble collapses, it again reaches its normal position. And through this we can sense if we have bubbles there or not. Of course, we can also, using data analysis, we can see that this dip can also be correlated with the size of the bubble. If the size of the bubble is exactly equal to the size of the nanopore, then blockage would be 100%. We won't see any current at that time. It means that signal would tend to 0. If we have a small nanobubble that doesn't cover the whole nanopore, or if we have bubble just above or below this nanopore, even in that case we can say that the channel is not 100% blocked. It means that we can in that case we can see that the dip in this current signal is not as large as in the case when all of the channel is blocked. So these are certain basic concepts for observation of nanobubbles. I think it's also better to discuss what is TM, because I will have certain questions from you and I want you to answer those questions, and or even not answer but think about those questions in this session. So in TM we use electrons as an illumination source. Now these electrons, if we use electrons in atmosphere, they'll ionize the gas or air molecules and very fast they will lose their energy, right? So what we want is that, let's suppose that we have an electron gun here and this is electron source, and we have a sample certain here, and then we have a CCD or a camera detector in the end. So this is a camera, this is your sample which you want to observe, and this is your illumination source or electron gun. Now if this total space is filled with air molecules, electrons, they will lose their energy very fast because they have very high wavelength. They will dissipate the energy at once. They will ionize the air molecules and they will lose their energy. So we must make sure that this whole space is kept at a very high vacuum. So it should be in high vacuum. High vacuum, so that electrons, they don't lose their energy. That is the one prerequisite for using the TEM. The other prerequisite is that the camera or the CCD is on the other side of the sample. It means that these electrons, they must pass through the sample and then hit the CCD here, right? So of course electrons, they will lose a lot of their energy here, but they shouldn't lose their 100% energy. It means that this sample should be ultra thin, and when I use the word ultra thin, it should be nanometer scales. So this sample must be ultra thin. Only then we can observe the image of this sample using a CCD camera that is below the sample, right? So this is the fundamental of using the TM here. Now briefly I'll talk about techniques and then we'll discuss. So I have here mentioned about 3 techniques. The first technique is called the liquid phase electron microscope. Liquid phase electron microscopy. In this technique, what we do is we observe the bubbles inside liquid. So, and this liquid is not frozen, it is in liquid state. So it means that we have to do some kind of nano fabrication so that we are able to encapsulate the liquid between 2 ultra thin films, right? So here what we are doing is that we have a silicon nitride membrane. This membrane thickness would be anywhere in the order of 25 to 50 nanometers. We have the same membrane on the top side, and these 2 membranes are spaced using a spacer. So we have a spacer, its height will depend on how thick channel we want. So let's suppose that this spacer is 150 nanometers. It means that the liquid thickness in this channel would be around 150 nanometers. And then when we fill this spacer or this space between these 2 films with water, and then we irradiate the sample using an electron beam. But the question is, from where does the nanobubbles come then? Because we are just observing liquid. So these nanobubbles, if we provide sufficiently high electron voltage of electrons there, radiolysis might occur. And this radiolysis, in this radiolysis process, there are a lot of radicals which are being produced, and water is being dissociated into several ions and several species. But hydrogen is one of the species which is being produced there. So when this liquid, the saturation of hydrogen is reached inside this liquid, the solubility is totally saturated, then these bubbles, the hydrogen molecules, they start to agglomerate on the surface, and then the nucleation takes place here. And this all we are able to observe using a TEM. So this technique is called the liquid phase electron microscopy. The other technique is kind of a freeze method. and for this we require a special class of TMs that is called a cryo-TM. They work at very low temperature. So what we do is that we prepare a sample in such a way that first our sample is in liquid state, then using certain methods like ion exchange method or solvent exchange method we can form nanobubbles, and then we dip the sample inside liquid nitrogen. So it freezes instantly. And then when we observe this sample in the TEM, in a conventional TEM, in that case we can see these bubbles inside the cryo TEM, and this here is the nanobubbles. And as you can see that we can see several cracks as well, because during freezing there might be formation of certain cracks along the ice crystals. This is another method. This paper was published in 2012 by the group of C.D. Ohl. Here they have used the total internal reflection fluorescence microscopy. So what they are doing is that we know that the TIR, the total internal reflection, it takes place at the interfaces. And if at interface we change the density somewhat, we can get a signature, like, because it will have effect on the TIR, how much total internal reflection is taking place at that time. So what he is doing, he is using a laser, and from one side it is glass, and on this side he is able to produce nanobubbles using the solvent exchange method. And when the lights, when this laser strikes here, you see a change in the total internal reflection signal, and from that they are able to get this image. Of course, they are also using the fluorescent molecules which emit light when certain wavelength is being thrown onto that bubbles. Now one by one, or basically let's this video, I think someone was asking me the question if these video is of simulations or if this is experiments. So this video here is from experiments. So here we are using the technique of liquid phase electron microscopy, and the length scale, if I can, I think, show it somewhere. The length scale, this watch is 200 nanometer. So these bubbles are typically in the order of 50-60 nanometer size. Also, I often get this question if this video is in real time or are we using a high speed camera. So of course, this is not a high speed camera. In fact, we have slowed or we have fast this video. So this video is played, I think the time here is 1 minute 45 seconds. So this seconds video is for data for 1 minute 45 seconds. So this again confirms that these bubbles, they are very stable, and because this sample is continuously being irradiated by the electron beam, so saturation is continuously increasing. So that's why these bubbles, they are growing in size. Right. In the sensing based techniques, we talk about AFM and the resistive pulse sensing. So this is the image from AFM. Here again the bubbles, they are formed using the solvent exchange method. Now what is the solvent exchange method? In the solvent exchange method, what you do is that you take a liquid which has, in which the gas solubility is low, such as you take acetone or ethanol, and then you supersaturate that liquid with the particular gas. Then you put it on a substrate. In this case it might be HOPG. HOPG is a highly oriented graphite, and then you exchange it with water. Now what happens that when you exchange this ethanol, which is highly supersaturated with gases, with water, then these gases, they tend to stick to the surface, and when it is being replaced by water, the bubbles are formed because water is already supersaturated with the gases as well. So water cannot dissolve more gases, and then in that case we are able to observe these nanobubbles. And this image is taken by AFM by one of my friend Teshima, and you can see that he has observed the time after which these bubbles, how these bubbles behave. So you can see that he has observed this for 230 minutes, around 4 hours, and the size of these bubbles is typically around 500 nanometer. Yeah, this is 1 micrometer, so typically around 500 nanometer the size of these bubbles are, and all of these are gaseous bubbles. So we have seen that how these bubbles have evolved over time. This is one image of the AFM, which is used to observe or to sense basically bubbles using a probe. and I think this I have already discussed. So this is the kind of a CAD image where we have a nanopore, and this is a silicon nitride membrane, and on this we have a nanopore. I think the size which was used by Dr. Paul in this experiment was around 280 nanometers pore. And when we had a NaCl solution, and when we apply the voltage across these nanopores, we see the current signal as a variation of time, and you see a lot of drops. It means that at these times the bubbles are nucleated. Some of these signals, they reach to zero. It means that channel is totally blocked. Some stay at a certain distance from the zero value. It means that we still have some certain space for this electrolyte to flow. Now let us go into, let us discuss more about liquid phase electron microscope. So as we discussed in this lecture, that it has two major requirements. One is that the sample should have encapsulated water. It means that there should be an interface between the vacuum and liquid. So let's suppose that this is your, here we have water, here we have vacuum. And other requirement is that sample should be very thin. And in the beginning, I have told you that we don't, or we won't be using silicon nitride membranes, which I just discussed in the last slides, that we have silicon nitride membrane and we encapsulate water between two membranes. Now I want you to think of other options which can be employed for this, to encapsulate water and then observe. So please think about it, and maybe we can have a discussion. So what other materials can be used? That is the question. Can you think of any 2D material? Graphene is a 2D material. Sorry? Yes, that is also one of that. Hexagonal boronitride, HBN. So although it sounds like it is a very difficult method, but it's very straight method. We can of course use single layer or few layer graphene to encapsulate water, right? And that is one of the cheapest methods, because if you want to go for silicon nitride, you have two options, whether to buy it from market or whether you can have to CVD grow the silicon nitride on the substrate, and then you have to do the back etching. So let's say that in silicon nitride, if you want to do in-house experiments, you start with a silicon nitride, you start with silicon this wafer, you deposit on top of it some silicon nitride, and because silicon wafer is typically thick, it is around thickness of around 500 micrometer or somewhat in that order. So you need to remove, and the silicon nitride is, let's say, 100 nanometers thickness. So you need to remove silicon from here, so that the electron beams, they don't lose their energy in silicon. So that is a wet etching method. That's a very complex method. So usually what you can prefer is you can use few layer graphene. And for using few layer graphene, what you can do is, let's suppose when you go out in industry, you buy graphene which is already deposited on the copper substrate. So we start with a copper substrate. This copper substrate is typically 30 micrometer in length, thickness, and then we have a single layer or few layer graphene on top of it. Right? And the main thinking here is that if we can in the end make something like of this sort, where we have graphene here, and between them we have water. Of course, this is a misrepresentation of how the water is observed. usually water is present in pockets because these graphene, they have very strong van der Waals forces. So once they get stick to each other, it cannot be separated. So usually what we have is that when you observe inside a TEM, usually we get some sort of this kind of regions. So these are called water pockets. And then these water pockets is what you observe using the TEM. Now, how do we reach from here to here? At one stage, we have a simple fuel graphene on copper substrate, and on the other side, we have water encapsulated between the graphene layers. So what we need to do is, I'll of course show it using the sketching here. So we typically first have to start with the TM grids. So these TM grids, they are OEM available, and this is a mesh, and on this mesh we have a films of any material. Let's say we have a holy carbon film, and the radius of this grid is typically 3.05 millimeters, and it is not as expensive as a silicon nitride membranes. And then on top of it we have one holy carbon film. So we basically have a carbon film on top of it. This is amorphous carbon, and it has holes of certain sizes. First of all, what we need to do is we need to put these films or these TM grids on top of this fuel layer graphene copper substrate, and then using the van der Waals forces, it is expected that this graphene will stick to this film or this TM grid. So we are placing this grid on one side is holy carbon film on top of this singular graphene, and then we let it stick for a certain time. Then the next process should be to remove the copper, because we have to remove copper from here. Only then this can be transferred to this grid. So to remove copper, we can use any etchant, any solution which dissolves copper, and ammonium persulfate is such solution. So we dip these TM grids in the solution of ammonium persulfate, and once it turns blue, it means that all the copper is being dissolved now. So in the end what we get is, we get the TM grid, and if I zoom it, if I zoom one mesh of this TM grid, we have amorphous carbon film, and these carbon film is holy, it means it has bigger holes here, and inside these holes we have deposited the single layer graphene or fuel layer graphene now. This is SLG or fuel layer graphene. And after that, the method is very simple. What you need to do is you need to put a droplet of the solution which you want to observe. It can be liquid, it can be water, it can be any salts. It can, if you want to see any reactions which are being kind of happen due to electron beams or free electrons, that you can also see here, that you can also put here. And then once you prepare these two grids, you just sandwich them. You just put this grid on top of this grid. What it does is that these two, of course, when we put I'll just show you the side view. So this is our grid, and this is the droplet which we have on top of it. So when we put this grid on top of it, a sandwich will be formed, and due to the capillary forces, this grid, this water will evaporate, and this will try to pull it towards each other, and then we have liquid encapsulated between the two single layer graphene or fuel layer graphenes. And now this can be used inside the TEM, because now we don't have the water, it cannot leak inside TEM. So this is how you prepare a liquid cell. This is called a liquid cell. I would have loved to show you this liquid cells, how to prepare it, but right now we don't have that facility here. And This gives you so much flexibility that you can even observe how the self-assembly of certain molecules they take place, because we have such high resolution. So we can see the DNA. So using this technique, we can use we can even see the crystallization of crystals occurring. We can observe how NaCl is formed. Crystals of NaCl, you can exactly see those crystals here. So this is a very strong, powerful technique. And once this is formed, then if you observe this inside the TM, then you can see then because of radiolysis process, the hydrogen it splits into your H2 plus other free radicals and other species. And once the saturation is achieved, then we see nanobubbles nucleating inside this TM grids. So this is one of the ways using which we can observe nanobubbles. But this is mostly a skill-based technique. There are certain do's and don'ts which you have to take care when you perform this experiment. The first very critical step is that this copper is just 30 micrometer. It means it's very flexible. We can damage this very easily. So you must make sure that this is ultra flat, because we are here talking about a film which is just 1 or 2 atoms thick. Its thickness is just 0.3 nanometers. So this should be ultra flat, and you should use certain methods which you need to explore how to make it ultra flat. Maybe one is you can put it between 2 glass slides and apply a medium pressure so that it becomes flat. Then the next important step is that when you put this holy carbon grid on top of that, the holy carbon side should be facing downside. So there's enough van der Waals interactions between your holy carbon and this singular graphene so that it stick to it. Third very important step is that since it is a wet method, so you have to dip this inside a APS, which is a liquid, which is in liquid form, and in liquid there is surface tension is the main culprit. So this surface tension can damage the graphene films as well. So you have to put it very gently. A lot of control over hand is required, so that these grids they don't fail, or these graphene it doesn't get damaged, it doesn't get cracked or shattered in this process. Again, that kind of precision is required when you take it out again from the liquid, because again when we take it out, the surface tension it tries to pull it back inside, and that will again be a problem there. Then because we have etched away the copper in APS solution, so it might have certain impurities, certain copper deposits might be present there. Then you have to wash it using DI water for several times, maybe 3 hours, 5 hours. That is also one of the very critical step here that you must take care of. And in the end, when you put this when you sandwich 2 grids, it might happen that they are not aligned very well, and the TM holders which we have where this sample goes, they have exactly the 0.3 mm slot. If these 2 grids somewhat they are not aligned with each other, then they will not be able to fit into this. So that is other which you have to take care when you prepare this sample, right? So now what we need to do is that I want to take your inputs, that what do you think how we can improve this steps, of if we can innovate in certain materials which we can use here. So any comments based on this technique which you have so far? Yeah, clean room is required, but not like we don't require a like class 1000 or class these clean rooms. Maybe ISO 9 clean room would work here. So basically we want a dust-free environment, and we also want that not much air is in circulation there. That is what we require. But yeah, like I have seen people who have made this in clean room, and I've seen researchers who have made these cells in a normal lab without any clean room, and there's no much difference in the contamination levels. So that is one thing. I think one aspect or If you want to go into more details into that, I must recommend you, there's one video, of course we were not supposed to put videos here, so I haven't put that video here because that's not a copyright, but you can go in jove.com and here you just search liquid cell electron microscopy. You will see all the steps there. And I must also share that the TM is one instrument which has produced maximum number of Nobel Prizes so far. And this is one technique which hasn't got Nobel Prize yet. So we have got Nobel Prize for the low temperature TMs. We have Nobel Prize for the TM itself, but for this we still haven't got. So maybe if innovations in this method is you can propose, then maybe next time or some other time you will get that achievement. Yes. Now let us also discuss about disadvantages of this method. So I have spoken a lot of good things about this method, but it's not the case that it is very good. Can you think of any shortcoming of this method? Can you think of anything on which we don't have any control of? Well, we don't have any control over how many water pockets will be formed there and what will be the size of water pockets there, right? That we don't have any control of, right? Also, let me draw it here. So I was talking about a holy carbon film. This film has holes in it. So what usually happens is that, let's suppose we are talking about, this is a TM grid. Of course, it has mesh. If I focus on just one mesh, then on this we have holy carbon film and it will have certain holes, right? Now, we know that this is here is amorphous carbon. And its thickness is somewhat around 50 nanometers, right? So usually where we observe the samples is when we sandwich it. So it means that we have two amorphous carbon films, and these two amorphous carbon films, they must overlap each other. It means that when I place this on top of this, what I should get is, maybe this is one hole, this is other hole, and then we have graphene on both, suspended graphene on both these holes, and this is the region where the thickness would be minimum, because on the other regions, in this region we will have at least amorphous carbon from outside of this. So we have amorphous carbon here as well, and we have amorphous carbon here as well. But the intersection here we don't have any amorphous carbon, we just have suspended graphene layer here. So this is the region where you get the maximum resolution. But can you imagine like placing holes which are 2 micrometer in diameter in such a way that we can get suspended graphene? That is another challenge from this technique. So it kind of runs on probability. Of course, the holes are such spaced that you will find a lot of intersection points, but it could happen that over those intersection points there is no water. Then your experiment fails in that case also. So these are all the technicalities which you must take care of when you perform this experiment. Certain other technicalities, yeah. One good merit about this case is that once you make this graphene water pockets, you can have multiple experiments, because the graphene is impermeable to liquid or gases. The experiments have proven that graphene is impermeable. So once these water pockets are formed, inside like the vacuum inside this TM is 10⁻⁻6 pascals, 10⁻⁸ pascals, such low vacuum, ultra low vacuum. Even in that case, this is stable. So this is merit that this can be used multiple times. So if you make one good liquid cell, you can expect somewhere around 500 water pockets in that. And it's difficult to observe all the 500 water pockets in one go. So you can maybe radiate one sample first and one section of sample first, observe the bubbles or any of the phenomena which you want to see, and then maybe switch to other in the next experiment. That's how you can do it. And yes, that was all about liquid phase electron microscopy. not liquid phase, but the graphene liquid cells. So here we were discussing about the graphene liquid cells. Right now, where research is going in this side, it's all about how to make cells where you can control the flow of liquid. So many researchers, they are trying to make hybrid liquid cells. In these hybrid liquid cells, what they do is that one layer is silicon nitride, one layer is graphene. The advantage of graphene is that it is very ultra thin. The silicon nitride is 20-50 nanometers, graphene is 0.3 nanometers. So you get a very high spatial resolution in graphene. So what they try to do is that maybe using the silicon nitride and etching, they make channels in the silicon nitride membrane, and then from top they cover it with single layer graphene. So using this also they are able to specify, or they are able to control where the liquid would be, because in water pockets we don't have any control, but here we have channels. So we can control the liquids, and we can use various MEMS device, various flow actuators to even flow liquid from it. So even flow is now also possible in liquid cells, where you can adjust the flow rate of reactants and then see how the reaction goes under various conditions. The other variation of these experiments is towards the TM holder, so where you hold the sample. So now it is also possible to heat the samples, so that you can see at what temperature, what is the effect on these bubbles. In my, of course, I have also done some experiments on temperature based these liquid cells, but what usually happens is that when we try to heat the samples, the flow of liquid due to convection is so fast that we are not able to observe anything. But there are certain papers where they have observed the effect of temperature on nanobubbles using the same technique. So that is where you can also innovate in this sector, in this domain. Yes. But remember that these bubbles, they are just the gas bubbles. They are not the vapor bubbles. So we have tried to heat, we have tried to fill the complete cell with liquid, and then we have tried to heat it, and we have tried to put the electron beam in such low configuration that radiolysis doesn't occur, so that all the vapors which are formed, all the bubbles which are formed, they are vapor bubbles. But that hasn't happened. What happens is that complete drying of the sample happens. So we are able to see the interface, but bubble never. But there was one paper long very back, 2013 paper, where what they have done is that they have tried to fabricate a nano heater here, and then using the nano heater, they have, when they supply the voltage here, they are able to see bubble nucleation happening at the same region again and again. So it means that they are able to observe it, but because nano bubble, these vapor bubbles, they are so fast, they are only able to get 2 or 3 images. What they expect is that grow, collapse, grow, collapse, grow, collapse. These gas ones they are pretty stable. So gas ones you can observe for a very long time, 2-3 hours they'll be there. And then when you adjust the radio electron beam, you can basically control the saturation level there. And then if you make the beam more intense, bubbles will grow. If you make the beam weaker, the bubbles may shrink. That all you can observe there. Yes. So this was about liquid phase electron microscopy. What I suggest, if you want to read more about it, there's a wonderful book by F.M. Ross. So F.M. Ross was one of the pioneering of this field. So she used to work with Intel, and in Intel she used to work on liquid cells. So there's a book on, the name is Liquid Cell Electron Microscopy by F.M. Ross, and I think PDF is available online, so you can just have a look at that book, and a lot of things about this technique or the bubble formation is discussed there. That book, I think it came out in 2017, 2018. So any questions so far from this topic? So, of course, we can apply continuum based equations. Continuum is still valid here. I think if you want to apply the statistical based equation, that is in ones of nanometers. Here we still have continuum based equations, so they are valid. So I think the continuum it breaks under 6-7 nanometers. Around for what 70 nanometers? Okay, okay. But what I've observed is that there are some groups who what they do is that they do the experiment on bubbles, then they also apply the COMSOL models on these bubbles. So continuum approach there. One thing I've missed here is that the bulging of these water pockets and water, because he asked about the continuum approach, so bulging also occurs here. So what do you mean by bulging is that, let's suppose that we have a liquid cell. This is silicon nitride windows. And then this is at atmospheric condition. But when we put this inside a TEM, which is at a very high vacuum, and this is at atmosphere pressure, so the bulging can occur here. It means that these films they bulge outwards, which then further reduce the pressure of these liquid cells. But it is difficult to quantify how much bulging is observed inside a TM. But there are certain groups who have tried to deposit the same thickness of liquid, silicon nitride films onto a substrate, and then at certain pressure they have used the AFM to see how much bulging occurs. So bulging typically it can range. So for a liquid cell whose spacer thickness is around 150 nanometer, this bulging can occur in the scales of around 500 nanometer. It means that the thickness it can go up to 3 times more. So if you have a liquid cell where you can pump in more water, or which is open to the ambient, in that case you can still fill this full cell and make sure that the pressure is atmospheric. But if you are dealing with the graphene liquid cells here, in that case we cannot fill the liquid again. So that would be at lower pressures. Yes. So any more questions? All right. So let us now discuss briefly about AFM based technique, and then we'll move toward the resistive pulse sensing technique. So AFM it is also called as the atomic force microscope. And as I mentioned earlier, that it also works on the principle that we have a very thin tip, and that tip it raster scans onto the surface, and we know that as the distance becomes very small, the force or repulsion it becomes very strong. Sorry, it is somewhere like this. Right? So usually we operate in the domain where, because we are using tapping mode method. Tapping mode means that the tip it kind of taps onto the surface and then as it raster scans. So since these bubbles they are soft matter bubbles, so in this case it is important to see on what modes we work. We work on the amplitude mode or the frequency mode, and we get images such as the profile of the substrate. Now in the case of TM, the reason of the bubble formation was that we were using electron beam, and that electron beam it radialized the water, and from that when the hydrogen molecules are produced, then they kind of agglomerate on the surface, and then we observe the nanobubbles there. But in this case we are not using the electron beam. So what could be the reason for nanobubble formation? So here what they do the solvent exchange method. So let's suppose that we have a substrate, any hydrophobic substrate. Hydrophobic substrate means that the surface energy of the solid vapor would be less than that of solid liquid. So it would have more affinity towards to stick gas on top of it than liquid, right? So first they take a solvent or a liquid such as acetone or ethanol, and they supersaturate this with gas of which bubbles they want to see. Let's suppose they saturate it with nitrogen gas, and then they place the substrate in this liquid for certain time, and then they, let's suppose that we have set up as we have a petri dish here, and in this petri dish we have a substrate, and this is hydrophobic, and this is totally immersed in this acetone or ethanol, which is supersaturated with gas molecules, nitrogen gas, and then we replace this liquid with water at atmosphere that already have nitrogen dissolved into it. So what this leads to is that as the acetone is replaced, and water, it cannot take in more gas, so these bubbles, they start to form here. Sometimes these are bubbles, sometimes these are pancakes. So usually on HOPG we refer them as pancakes. Pancakes because they are ultra flat. So the thickness, it might be somewhere, I think I can also show you with this image. Yes, so you can see with this image that although the lateral distance is around 500 nanometers, but the thickness is just 40 nanometers. So the contrast is 1:10. It is ultra flat. So these solvent exchange nanobubbles are formed. In the case of silicon nitride, which is a hydrophilic membrane, we usually see the bubbles having a very high contact angle because there the saturation is very high. But here saturation is low, so kind of the molecules, they stick to the surface, and then we kind of see a pancake. So although in this image you can see that bubbles are spherical, but if you see the in actual scale, it must be somewhat like this. This is how the pancakes they look. Yes, so then these bubbles of pancakes are formed, and then we put the same sample inside. So since this is a wet sample, so we have to put it with the petri dish itself. So we put the sample inside a dish, we have water filled in it, and then we have bubbles, and then we use the AFM to kind of raster scan this surface, and we get both the lateral as well as the height of these bubbles. One advantage of using this AFM method is that it gives us a 3D picture. In the case of TM, we just able to get a 2D image, and we need to do a lot of analysis to extract little bit data about the height profile. Of course, we cannot find out what is the exact height, but we are able to trace the profile based on how much is the intensity of electrons at each pixel. But in this case, we are able to get both the lateral and as well as the height of the bubbles. That is one advantage of this method. And this was one of the first methods which kind of found nanobubbles. All the liquid based method, they came later on in 2010 afterwards. This method was, I think, first used in 2003 or 2004 to observe nanobubbles for the first time. So this was all about this AFM based methods. Now let us also discuss about the sensing based method or the resistive pulse sensing technique. In the resistive pulse sensing technique, yeah, let's first discuss about the signal processing. So for that, we are not going to use bubbles, but we are going to use the DNA to see how the strands can be detected using this pulse sensing technique. So of course, I've mentioned this before. So we have a nanopore, and then we have a tanks. These tanks has electrodes, and it is filled with, let's suppose for first case we fill it with DNAs. So we have a lot of DNA strands, helical strands of DNAs, right? Now these DNAs, they have nucleotides which has certain signals, the AGTL nucleotides. If you see the structure of DNA, it has nucleotides which give you the DNA sequence of any subject. Each person, they have a unique DNA sequence. So these nucleotides, they give the DNA sequence. And these nucleotides, they have certain size, certain physical parameters. If we are able to make a very small, tiny nanopore, and then the DNA, it passes through this nanopore, the amount of signal blockages which we get, both the duration of the signal blockage as well as the depth of the signal blockage, that can be used to find what sequencing we have in the DNA. So this is the beauty of resistive pulse sensing technique, that although we are not able to see any phenomena happening there, but we can still get signals which might be able to use to sequence the DNA. And DNA sequencing is one of the hottest market right now, because if you are able to, Hanji, yes. So this should has both a very wide bandwidth, and at the same time it should be very sensitive. So it might be able to get the current in the scales of nano amperes, and the time scale should be in gigahertz. Because although I'm this time here, we can have hundreds of signals in 1 millisecond time or 1 microsecond time. So this should be in gigahertz sampling rate we have. Yeah, so I was discussing that this DNA market, it's kind of very popular, because if we are able to control the DNA sequence of any human, any subject, then we can able to control the disease it might occur in the later stages, that Alzheimer's or any heart disease that can be that can be controlled by rightly sequencing or rightly treating the DNA. So a lot of work is going towards this field. This is one of the very exotic application of this resistive pulse sensing technique. But again, this can be used to see something very basic like vapor nanobubble. So in the case of when we observe vapor nanobubbles through this, so we fill this with electrolyte, and because of the flow of charge in opposite directions, a lot of heat generation takes place, and then we observe the bubble nucleation. So in this case also we have got a lot of simulation results. There's a open software called ARB. It was developed in a university in Australia, and there they have the in-house code for this heat generation in a nanopore. And they also use they don't go for any molecular scales, they use the continuum models here, and then they are able to see how much heat is being generated inside a nanopore, and that sometimes goes all the way up till the limit of your homogeneous nucleation. So temperatures in scales of 590 Kelvin is also accessible in these small systems. So if you do the scale analysis, and of course there is a lot of losses when we talk about scale. Like they were news that we can reach to moon using CNT. If we do that type of analysis in this kind of nanopores, these nanopores, a multiple array can run the whole heat of the world which is required. That is the heat generation or volumetric heat generation in these systems. Because the volume is so small, the heat, even if it's in micro joules, it can be scaled because the size of the system is very small. But of course, when it comes to applications, it is difficult to realize that scales, because as we increase the number of these units, the losses, it also scales at the same rate. Yes. So Uh, I think that is all which I wanted to discuss about the experimental techniques for observing gas and vapor nanobubbles. I wish I could have used some videos here, but again, we are prohibited. But after this class, maybe we can have more discussions off camera about these topics. So, Manohar, you have some question?
Sir, can we, if we want to see these things through simulation,
Yes.
can we use software like Ansys or
So depend. See, ANSYS also works on the same. It also works on the continuum approach. So of course you can use it, but certain models which are fit for this that you have to use in those. So you might need to write certain UDF functions, but you can use that. So I know that researchers they use COMSOL for this kind of systems. ANSYS I haven't seen yet, but I don't see any problem of using ANSYS for these kind of simulations. Hmm? That I'm not very sure of.
Sir, one more question. You told that we can use X-rays are UV rays. So can we use laser?
So lasers also have the limit of wavelengths. So if we want to talk about the lasers, they are even beyond this light. So lasers are, if it is a red laser, it is around 780 nanometers, 1500 nanometers. So lasers like wavelength is also in the scale of nanometers. So of course, if you want to use laser, we have to see certain aspects of lasers. Like in the experiment which I which I showed in the one of the slides, where they are using a total internal reflection. There they are using laser, but then the main principle is total internal reflection. That is a very difficult thing. So once the nanobubbles is formed, it remains stable for a very long time. Of course, one of the methods could be if you pump some liquid from it which is having lower concentration of gases into it. So boil water multiple times and then pass through it. By diffusion can, yeah. But we haven't tried that yet. In our experiments, we were using the normal DI water, which has having the solubility with the it was in the ambient. So in that case, when we pump in, even when we pumped the water, we were not able to remove the bubbles.
So when we go for a simple X-ray or MRI scan, is there a possibility that some kind of nanobubble is generated in our body also? So that kind of energy is not.
I think in X-rays we don't have that kind of energy. X-rays they don't go for radiolysis. It's because electron beam they have free electrons, then they go for radiolysis. The X-ray they don't have this kind of. In that case, X-rays are quite safe as compared to electron beam. Of course, electrons has ionizing effect, but this bubbles cannot form. Hmm? Thermal signals means? How to thermal signal? I'm not getting it.
What is the temperature change across the time in this?
What is the temperature across the?
Nanobubble area, contact area.
So how to do it experimentally? I think the bubble is so small that it is difficult to do the sensing, but yeah, I have seen. So there was a lot of debate that this liquid cells, this electron beam, they also produce heat. So I know of certain experiments when they have embedded the sensors inside the silicon nitride membrane, and they have tried to see how is the variation of temperature inside the silicon nitride membrane. So of course experiments are there, but I'm not very sure of how they embed it. Of course they have to do a lot of depositions, CVD, these things. But right now I'm not able to remember what they did. But I can share that work with you. That work was published in Micron, the Willis Micro or Micron journal, and that work was from I think somewhere from Europe. So they have tried to see because there's a lot of debate and also it's beyond measurement that if the electrons they strike the water if there are any temperature changes or not. So right now what we believe is that all the nucleation that takes place because of the oversaturation.
So sir, what is your opinion that deposition of these nanobubbles shall affect this sleep?
Yes.
That affects.
Yes, that it affects. That's been proven. So they have proved that's proven in experiments that in a microchannel they have produced bubbles on the sides and then they have tried to measure the slip. So
In the last method, the sensing method, smaller bubbles or because the position is off from the place we are sensing? Is it?
How do you know that? So yeah, so these experiments standalone, they don't say anything. We also have to study theory. So see, that is a very good question actually. So there can be multiple cases. Like let's suppose that we have a nanopore here, and we see a dip, and that dip is absolute. That dip is kind of a partial dip. So there can be two, three circumstances. One is that this bubble is here, still we have some space on top of it. The other thing is that it can happen that although this bubble was small in the beginning, it has popped out of nanopore, and that bubble is right now somewhere here, sitting here. So when we get these kind of things, we have to analyze that which out of these two is more probable to happen. But with surety, we cannot say what could have happened because this is not an observation-based technique. But one thing certain is that this is bubble because if you go for simulations, you can see the temperature is very high. This temperature is close to spin-order limit, 574, 580 degrees, Kelvin temperature. So these under certain temperatures, even we have so much confinement, we expect bubbles to grow. But observation is still a problem. So when I last followed this work, I know that they were trying to remodel a old SEM, which was not in use, so that they can also observe or see these vapor nanobubbles. So they were trying to fit this system inside an SEM and then observe the bubble nucleation there. But that was not yet completed till the last time I heard about it. If you want to explore more about this field, so I would suggest some papers to you also. So have a look at those papers because this nanopore thing, it is the literature is very limited. So it was first taken by some group in Israel, and then in Tokyo, the group was working on it. But this is a very powerful method to observe vapor because vapor is difficult. Gas we can observe using AFM, you can observe it using TM, but vapor bubbles, that is the difficulty. And also one more thing is that vapor nanobubble, it's the probability of occurrence of the homogeneous nucleation is very difficult. But in such confinements, we observe homogeneous nucleation.
So that bubble, once that is generated at the junction of the nanopore, that collapse also?
Yes, it collapses. It also fluctuates.
It fluctuates also.
It also fluctuates also. So
something like chemoluminescence, etc. That kind of things.
Sorry.
Sonoluminescence or something like that.
So sonoluminescence can also occur, but we have not observed that because I think because of the size of the bubble is so small that the light detector which should be used to detect the sonoluminescence, it has to be very sensitive.
The temperatures which you were suggesting is of the order of the temperature of the sun.
Temperature is very high.
Temperature is
Extremely high. Temperature is it's 574 Kelvin.
somewhere around 6,000 Kelvin or something.
No, 574 Kelvin. 600 Kelvin.
600.
That is not high. But if you reduce the size of the bubble, reduce the size of the pore, then that temperature can be accessible.
So you were reporting about some kind of chamber which was made and there was a nanopore in the So what was the length or size of what? Is it possible to fabricate?
There they used to use the method of 3D printing.
3D printing. So it's a macroscopic scale.
It's a macroscopic scale. That
Nanopore.
they used to do milling, the gallium ion milling. So using SEM they used to do the milling, and this milling we have a sequence of gallium ions that bombard the silicon nitride membrane, and they strike out atom by atom, and then we have this pore. So also this pore, because although we are seeing it to be a straight line, like very thin, but this membrane also has thickness of around 19 nanometers. So it's kind of a channel in one aspect. The ratio is 1:4. So its alignment is also very difficult.
Such facility of creating such holes is not present.
Over here we have.
Over here.
We have the gallium ion mills in the C4D FED, and there we can in IIT Mandi we can make certain these holes.
And what kind of voltage was actually given to this chamber? So is it DC voltage somewhere 9.
हाँ,
Or it was
DC
very
DC
high.
voltage, and no, not very high voltage. I don't remember exactly right now. I think it was 6 or 9 volt, but I don't remember exactly. I think after this we can just have a. I will just show you the reference and then we can have.
Because this appears very fascinating.
This appears very fascinating, and the good thing is that this experiment is very modular. You don't need very big setups for that. So I remember the researcher who used to work on this topic. He used to carry his set in a very small box, and yet producing so powerful results.
But after the generation of this nanobubble, can we actually take it out, or maybe, or it is just for the demonstration purpose?
So what do you want to take it out?
Once nanobubble is generated,
हाँ.
so can we make it some kind of
So
continuous flow kind of device?
so the ultimate aim was to make a nano bubble emitter.
Emitter.
So nanobubbles. So you see the problem in these microchannels is that once the phase change occurs, the pinning occurs of the bubbles, and it causes dry out in the microchannels. So if we are able to produce homogeneous bubbles in the first place, then the length can be optimized so that we don't have the problem of pinning and these dry outs there. So that was the main idea behind this work, to make a nanobubble emitter.
So some companies actually suggest they are using these nanobubbles for water purification and all.
हाँ.
They are mechanically generating.
They are mechanically generating, I think. I have not followed their work, but I happen to visit some papers. They use a very simple method. I think I was discussing. So they are using the injection syringe. They block it from the front so that it becomes an isolated system. Then they do multiple pumpings. So they say that produces nanobubbles, but that could be the gases which are dissolved.
So what we used to do, we used to actually separate the ores. So there is a process called growth process.
Yes, we have.
We used to use some kind of porous membrane, and we used to push air through the porous membrane.
Okay.
That actually used to generate a large bubbles.
Bubbles. Okay.
Sizes we haven't actually seen.
Okay.
So
Okay.
since what you are suggesting is there is a simple syringe there
हाँ.
because of the very high pressure drop.
Right.
people are able to-
So I'll show you the paper also. I think that paper was there in the first slide. I think we can end this lecture. You know? So I think we can end this lecture and then have discussion of.